Well, good afternoon, everyone, and welcome to MyOrigin, Take Your Practice to the Next Level webinar with Dr. Scott Bergman. Dr. Bergman is a chiropractor and a board-certified naturopath and certified functional medicine practitioner. Dr. Bergman is currently the director of Chirokinetics, an integrative health clinic combining chiropractic care, rehabilitation, Pilates, core yoga therapy, and functional medicine. And today we're excited to have Dr. Bergman. He's going to focus on the detox panel of the MyOrigin system and how we can incorporate that into our practice. So just as some good housekeeping, we're going to have a 40-minute webinar with a 20-minute question and answer. So please reserve your questions to the end. You'll see in your GoToWebinar panel that you can raise your hand. So at the end, you can raise your hand and I can unmute your mic and then you can ask your question directly, or you can actually type your questions right in into the system and we can answer them as we go. So without further ado, let's welcome Dr. Bergman.
Thanks, Joel. Hi, everyone. Again, my name is Scott Bergman. I know how busy everyone is, and I really want to thank you for taking the time out of your day to further investigate how you can help your patients out. We'll be talking about the detox panel from MyOrigins, and hopefully we'll get through. Oh, trying to move. There we go. So here's our objectives for today. Although most people might already know this, we're going to talk a little bit about genetics, do a little bit of getting us all on the same page about genes and chromosomes and SNPs. We'll talk about the testing terminology, as well as looking at some gene functions, how the enzyme functions, specifically for the liver, the gut, and estrogen detoxification, how to navigate the MyOrigin report, the terminology and interpretation, and then what I use for applying the findings into protocols and looking at the gene variations and how I explain things to patients. So as we move forward, there we go. I don't know if people have heard Jeff Bland talk about his Book of Life story. I think it's probably appropriate for us to start there and about genetics. We know that we've got 23 pairs of chromosomes, 46 in total. Half come from the egg and half come from sperm, and that makes us what we are. So when we look at this Book of Life, and this is how I explain things to our patients as well, is the book is like a physiology book that we've all read in school. The book is you. And when we open that book up, there are sections. And in those sections, we're looking at different systems, whether it's the cardiovascular system or musculoskeletal system. As we look at the chapters, we look at organs and tissues and cells. But as we start getting into the specific pages, that's analogous to the chromosomes. The paragraphs could be seen as the genes. And as we drill down further and further, the actual letters of the words on paper in our book are the nucleotides. If you were to open up your book at page 250 and read those words, and then close that book 10 years later, if you were to open up at page 250 again, the words are going to be exactly the same. So the genes are what is written in ink, and they do not change. I'm going to try to move forward here. When we look at gene function, the genes themselves help produce polypeptides. So they help produce our receptors, our enzymes for conversion. Proteins are built. A whole bunch of things have to occur in order for that to go right. There has to be an uncoiling of the DNA. There has to be a copying of the information. Those specific words have to get copied, and then from transcription, they get translated into specific proteins. And then over time, we actually have to have some enzymes that degrade the systems that translate. So there's a lot going on a moment-to-moment basis of uncoiling the genes, copying, transcribing, producing proteins, having those proteins go to the specific cells, whether they're carrier molecules or they're there just for rebuilding tissue. A lot of times when we look at the reports, we're trying to, it can be confusing, the difference between an allele and a SNP. So I wanted to make sure that we went over that. An allele is a specific gene that has an alteration. So an allele is an alteration, and it can be one of two or more alternative forms of that specific gene. A SNP, a single nucleotide polymorphism, is the alteration. So it is the specific alteration within that gene. So those are the differences. Some people will say allele, some people will say SNP. Just realize that allele means that there is an alteration, SNP is the actual alteration. And when we talk about those alterations, an example of that would be if we had something like a cytosine, there's a specific shape, and that will always bind to a guanine. So if we see a nucleotide, that could appear differently on individuals. So if there is a change where that location is, so instead of having a C, there might be an adenine and a thymine, just that one change of this nucleotide, that creates the SNP. So instead of having a CG in that codon, which is a set of 3 nucleotides, it's an AT at that location of the codon. So that would be the specific SNP. So the SNPs are a swap at a specific rung location of that DNA ladder, and you'll hear words like or see things like heterozygous or homozygous. Heterozygous means that one set of the chromosome is positive, meaning that there is a variation, and the other set does not have a variation. So it's different. One set is positive, one set is negative. That means it's different. Heterozygous. Homozygous could be either a minus or a plus. So on the reports, minus means that there is no mutation or variation or SNP. It means that it is normal. Plus, which is also homozygous, will mean that from both sets of chromosomes, there are alterations. And so when we look at the report, you might see something like the MTHFR SNP at that specific location that will either be normal, might have one variation, or 2 variations. So the big question is, why should we care? What SNPs will do, what these variations or mutations will do, they're either going to slow down receptor and carrier protein function. They can slow down the production of enzymes, or they may speed up specific enzymes. The other thing that it can do is, because there is a variation at those SNPs, when there is translation and replication, you can actually convert to a completely different amino acid compound. So where there might have been, let's say, something like a cytosine, it could be turned into a valine, or actually more accurately, alanine could turn to a valine just with a specific SNP. So now you have different amino acids that are being produced that can alter function down the line. Some of the research, specifically with MTHFR, they are showing that a heterozygous plus minus SNP can reduce the ability of methylation by 30%. Optimal conversion of about 70%. With a homozygous plus plus, someone can have an alteration reducing their function to only 30 to 50% of optimal methylation, and we'll see why that's a big deal. So converting this 5,10-methylene tetrahydrofolate to the usable form of folate for proper methylation, the 5-methyl tetrahydrofolate, this can be reduced anywhere from 50 to 70% with a plus plus. Now, although the Book of Life is the words printed in ink and it never changes, we also have the epigenetics, which is the interpretation of that book. And that interpretation can change over time based on diet, lifestyle, exercise, our mental well-being, and environment. I actually use the acronym diet, exercise, attitude, lifestyle, environment. That's your deal. So when we talk to the patients about epigenetics, I ask them what their deal is. And we look at those factors. And the National Library of Medicine determined that 75% of our health and lifespan is determined by our epigenetics, which is the interpretation of our gene expression or suppression. So following the Book of Life analogy, there are sticky notes and there are paper clips. And we can put sticky notes on certain aspects of our Book of Life so that we can thumb right to that area, open the book, and express the words that are on those pages. As well, if we know what our genetic makeup is, we can put paper clips over certain areas of the book, maybe 5 or 6 pages that have to do with atherosclerosis or insulin resistance, so that when we're thumbing through our Book of Life, that paper clip closes all of those pages and you skip right over it. So that's the change of interpretation. And another analogy for a book is I remember reading Julius Caesar, right, Shakespeare, and not having one clue what was going on in that book. I didn't understand the characters. I didn't understand the language, how it was written. I didn't understand the importance of it. Didn't understand one thing. Well, my son was reading Julius Caesar a couple months ago, and I was reading through it, and as I was reading it, it had a completely different application than it did when I was in high school. I kind of understood it. I understood the characters. I understood the theme. I understood even how it related to current events in today. So that's an example that I use for people where you read a book, the words are the same, but how we interpret that book later in life is very, very different. So that's our epigenetics. And one thing to be clear is that we don't treat SNPs. right? You can't treat something that is always going to be that way. You're not going to change the SNPs. They are not diagnoses, but they are guideposts. And what they're capable of doing for us is identifying potential long-term altered function. And as practitioners, we're then able to understand that if there's a 3 or 4 or 5 step process for methylation, and we can't convert folic acid to that 5-methyl, we know that we can support those methylation pathways by just giving that usable form. Same thing with the B6. If we're not able to convert regular B6 to the paradoxal form of P5P for amino acid formation, just by giving a little P5P will change someone's ability to build muscle and to help the adrenals out. So helping with those pathways, looking at certain nutrient requirements, and most importantly for me, the way I look at it is supporting the nutrient reserve deficiencies. When someone has, let's say, the MTHFR, and if they're only methylating at 50%, the body is under a greater strain to methylate than someone who doesn't have that variant. So they require more magnesium. They're going to obviously require more 5-methyl as well. So looking at where the body is struggling, we're going to realize that their requirements for certain nutrients are going to be higher than their possible intake, which is where the supplementation and the protocols come from. So I speak to my patients about what I call NERD, nutrient reserve deficiencies. There's physical, chemical, which genetics would fall into that, and emotional stress, and all of those deplete nutrients. And that stress also increases the need for those nutrients. There's an increased stress on the organs if they don't have the nutrients they need, and that can lower their immune defenses. There's 68,000 ICD-10 codes. All of the chronic conditions can be associated with at least one nutrient reserve deficiency. Whether it's in the liver or whether it's in the muscle or in the organs. So we're using these macro and micronutrients 24 hours a day, and if those nutrients aren't there in the system, especially because of a genetic SNP that requires more nutrients to get the same job done. If we're working at 40% capacity methylation, our methylation process essentially has to work 2.5 times harder to get the same job done. If we're working 2.5 times harder, we need 2.5 times the nutrients, certainly above RDA levels, right? So one of the things that I looked at was that organs with adequate reserves have a greater resistance to stress and endogenous and exogenous toxins. Every single organ has their primary function and secondary function. The liver has over 360 functions, but one common function that all the organs have are as nutrient reserve storage sites. So for bone, when I talk to my patients about NERD, I'll say, what mineral do you think stores in the bone? And they all know that it's calcium. And when I ask them what condition comes from bone or calcium not being in the bone, and they all know that it's osteoporosis. Well, as the body needs more calcium, if we're not taking it in, and the body needs that calcium for simple things like just muscle contraction, like the heart needs to contract. As calcium levels get depleted, the body will just pull calcium from the bone so that our heart keeps beating. That's a good thing. That keeps us from dying. However, it makes the bone more porous, and that then degrades the health and well-being of the bone. So the nutrients keep the organs well, and the organs use themselves as storage facilities for specific nutrients. So we know that like the liver stores vitamin A, D, E, and K, the fat solubles. Also, it's a storage site for B12 and B9 and iron. We talked about calcium in the bone. 99% of the calcium is stored in the bone, but also 60% of our magnesium is stored in the bone. So if we're magnesium deficient because of a specific SNP, we can create an osteoporotic situation because the body continues the need to pull magnesium out of the bone. And obviously muscle stores glycogen, and the electrolytes need to keep our blood buffered so that we have a proper pH, and needs to keep our muscles functioning, contracting. So all of these electrolytes can be pulled out of the bone or out of nutrient reserve sites to keep our bodies functioning as well as possible. Understanding our SNPs, we will understand what the propensity for nutrient reserve deficiency will be. So let's talk about detoxification itself. And again, just a quick review so that we're all on the same page. From a basic explanation of liver detox, we have phase 1 and phase 2 of detoxification. Phase 1 is taking the free radicals, the metabolites of our normal metabolism, and converting that semi-soluble metabolite into something much more toxic, much more reactive, and much more highly oxidated. And it's much more dangerous. And that happens in phase 1. In phase 2 of detoxification, we're taking that more reactive free radical and then surrounding it by a water molecule. And then from there, we're able to either pass it through the kidneys as urine, we're able to pass it through the stool. There's also respiration and perspiration, but mostly from detoxification, we get this pathway. So this is the basics of phase 1 and phase 2 of detox. Turning the page and going a little deeper, we have our total endogenous toxins, we have our total external toxins, our exotoxins, and those are non-polar and lipid soluble. And in our phase 1 detox, mostly through the cytochrome P450 family of enzymes, there's I think over 60 of those, we go through several chemical reactions: the oxidation, reduction, hydrolysis, hydration, and dehalogenation, which again can create these superoxide free radicals, and if not converted through phase 2 like we discussed, they could create tissue damage. In that intermediate phase, the molecules are more polar, more water soluble, but they're highly reactive intermediates, and the body requires these antioxidants to help with that intermediate stage. The more antioxidants we have, the easier we're able to transition from phase 1 detox to phase 2. And you see the list of enzymes and cofactors and nutrients needed for the cytochrome 450 enzymes to even be produced and convert over to those intermediates. As we move to the phase 2, the conjugation pathways, that's glutathione conjugation, amino acid conjugation, sulfation, glucuronidation, acetylation and methylation, the nutrients required are things like NLC, calcium D-glucarate, cysteine, methionine, glycine, all of those amino acids, and especially magnesium. So that brings us back to the need for those looking at NERD and the nutrient reserve deficiencies. Peeling back even further, we're looking now at the specific genes. And these genes are in, most of them are in your detox panel. So we'll see the cytochrome P1A, 1,2s. We'll be talking about the ones that are highlighted in a bit. And glutathione production and conjugation requires these genes here, and these are all in your detox panel as well. So if you see a variation, a SNP in any one of these genes, you'll have an idea of what those genes should be doing. A SNP will determine whether things are upregulated or downregulated. We'll talk about that as well. But to be able to know the difference between someone's inability to detox because of an estrogen detoxification versus a methylation or a gene expression or an inability to produce enough amino acids, I think is a big deal, because that changes protocol quite a bit. Maybe not in the beginning when we're doing a 4R program and we're just trying to clean things out, but for our patients in the long run, to provide them a wellness, preventative, a proactive care plan, I think anyway, it's very, very important to understand what the patient will always be struggling with. And by supporting that, we're going to be able to help them in the long run. Here's estrogen metabolism. And what I'm showing here are the genes and the enzymes. But just to create our total estrogen pool, our 17 beta hydroxys, all of this needs to take place. All of these chemical reactions. Just having a SNP in our ability to produce DHEA is going to determine how much aromatase we produce. And whether we have a SNP in our 19A1, that determines whether things are upregulated in our aromatase or downregulated. And most of the, well, all of the estrogen-based cancers, one of the common therapies is aromatase inhibitors. Well, instead of trying to force the aromatase to be inhibited, my goal has always been to try to move that estrogen pool into something, into a healthier pathway. Here are the 3 detoxification pathways for our estrogens. We've got, we have our green light estrogens, our yellow light estrogens, and our red light estrogens. And we can see here that CYP1A1, the CYP1A2, if there is a SNP in one of those genes, we're not going to be producing the enzymes necessary to encourage our total estrogen pool to go down the green conveyor belt to be methylated, to convert to the 2-methoxy estrones to give us the therapeutic value of estrogen. Or in our detox profile, if we're not methylating properly, or if we have a COMT issue, we're definitely not going to be able to move things down our green light. If we have an alteration with our 3A4 or the 3A5 or the C19, that's actually going to upregulate and kind of pull estrogen towards the 16-hydroxyestrogens, which we know are a little bit more inflammatory and will promote fibrocystic breast, endometriosis, PCOS. So we want to be able to reduce the amount of 16s and increase the 2s as much as possible. Running a 2:16 ratio is great to see what the estrogen levels are in the moment, but what we will not see is if there was a genetic propensity to promote a higher 2:16 ratio, right? How much of it was diet and lifestyle? How much of it is there's going to be a constant struggle of balancing those two out? And then if we have the CYP1B1, if there's a mutation there, that's an upregulation of that gene, where as well, it will force or encourage the estrogen pool to the 4-OHs. And if we're not methylating or have the COMT, well, then it definitely promotes increasing the estrogen quinones and being a possible carcinogen. That's all the stuff just for estrogen metabolism that we get from this detox panel. So I find that to be pretty fun to look at and pretty interesting. This is pretty much what I just discussed. When we look at the 1A1, we get a decrease of the 2-hydroxyestrogens. If we have a 1A2 that is off, that actually promotes the 16-OHs. Also decreases caffeine clearance. So if we keep caffeine in our system a little longer, that can create anxiety. So when you're looking at anxiety or depression, we can see the potential for that within the detox protocol. And I'll talk a little bit about some case studies later, but some pretty big eye-openers for the patient when they realize that some of this stuff is written in ink and those words can't be changed, but with proper lifestyle modification, things can be suppressed or expressed. It really motivates the patient to do the right thing for themselves. For an overview of the phase 2 conjugation pathways, so we know that glutathione is one of the most prevalent antioxidants in our body. It's a powerful family of antioxidants, the STs, GTs, SSs. If our body is not conjugating our amino acids properly, we're not producing enough glycine or taurine, so we can have a lower ability for protein synthesis or even collagen formation. So from a musculoskeletal point of view, I think that's pretty important. And we know that taurine helps with cognition and cell membrane health and electrolyte balance. I found that when I was speaking to my patients and telling them that they have to do a better job with their nutrient intake, once they understand that there's a genetic propensity to always need more, they seem to be much more compliant. On the detox pathway, there's also the SULTs, the sulfation, which are B6 dependent. So again, if we're having a problem with simply not getting enough B6, we're not going to be able to sulfation pathways will be inhibited. Sulfation is required for DHEA levels for adrenal fatigue. So I think that's an important marker for us to look at. Yes, we always want to provide DHEA or a 7-keto DHEA if we need to, maybe even a little progesterone to help with the adrenals, definitely B5, B6. But if we know that there's a genetic variant that DHEA is always going to be a little bit lower, then we're never going to be able to convert progesterone to testosterone, and then a proper conversion from testosterone to estrogen. We would start pushing those pro-inflammatory estrogen pathways just by not having enough DHEA. And if our DHEA is lower, we tend to have higher levels of cortisol. We can keep giving nutrients to suppress high cortisol, or we can look at the pathways of sulfation and try to improve those. So sulfation also metabolizes hormones and neurotransmitters. The glucuronidation, beta-glucuronidase is in our colon. That is a kind of a variant form or an improper form of glucuronidase, and it reconjugates estrogen. So if our glucuronidation isn't working properly, we get a higher beta-glucuronidase, and we get a higher reconjugation of estrogens back into our system, creating estrogen dominance. Glucuronidation also breaks down the bilirubin into a non-toxic compound, and to a point that we're able to clear that in our urine. So if we don't convert that bilirubin to a non-toxic compound, we're creating an inflammatory response. The NATs are acetylation. Acetylation is required for proper gene expression, and there are aspects of phase 2 required for within acetylation for phase 2 pathways. And then we have a bunch of methylators, the MTHFRs, MTRs, MTRRs, and the COMTs. All of those are in your detox panel to see how well we're methylating. Oops. And methylation is a big deal. It deserves its own slide. So just by having a SNP in any of those methylation promoting genes, we have the ability of reducing our energy, getting CoQ10 and carnitine into the Krebs cycle for the mitochondria. We get a reduced bile production. Methylation is required for the myelination of the nerves. As we demyelinate, we get into some pretty serious neurodegenerative autoimmune conditions. Cell membrane health, generating and degrading neurotransmitters, generating SAMe for as a natural MAO inhibitor. So if we're not methylating properly, we can get an increase in dopamine, we can get an increase in norepi and epinephrines. That can create a lot of anxiety. So methylation is required for just mood enhancement. From a pure phase 2 detox aspect, methylation helps with heavy metals as well. And the histamines are something to look at anytime someone has an allergy issue and whether they're methylating properly. So to the reports themselves, they're improving every day. They look really, really good right now. When you look at the health action plan of a detox panel, the first page you see is a table of contents, so you can flip to it very quickly if you want to look specifically at individual traits. There's a page or two about understanding your genes for the patient. It's a good aspect to hand to them. And then how the traits affect you. There's an overall looking at these genes and just saying, hey, at 1B1, there's a high impact. Definitely want to look at that, as opposed to looking at something like the NRF2s. So this is a great sneak peek at what's going on, or sometimes even just something to give to the patient. Sometimes that's all they need. Moving further into the health action plan, we see full pages on supplement recommendations, based on the SNPs that were high and medium impact. There are specific diet recommendations and lifestyle recommendations, and you can see based on a particular SNP, whether it's NAT or your inflammatory traits, oxidative stress, and specific lifestyle exercise recommendations. And then based on the particular trait as well, recommendations of other tests that you might want to take to look at the specific markers in real time. Then you're able to discuss with your patient, well, your CRP is actually pretty good now, but you're 40. We don't know what that's going to be like when you're 60. So giving you some specific nutrients to help now, we're going to avoid possibly some very dangerous inflammation pathways. Further into the report are actual trait details. So this is all one page. Looks at the particular genes. This is for the inflammation trait. Then they look at the specific genes that cause inflammation, what the SNP is, that the RS number is the specific SNP and the specific location of that gene, and what that variation is, a plus plus or a plus minus or even minus minus. And then the report shows you the impact or how important that may be to your health. A little bit of a summary of supplementation that might be the most important with the diet, lifestyle, and further testing on each and every page. So in the detox panel, there's also the glutathione traits, the sulfation traits, the oxidative stress traits, and on and on. So a page on each one of these, if you wanted to look at it that way. And patients tend to like this a little bit better because of the explanations. I don't tend to look at this one too much. It's just too many pages for me to flip through. I'll show you what I look at. From the inflammatory page, this is something that I'll always point out to the patient or make little notes. TNF alpha, when there's a plus plus, that'll upregulate the level of inflammation. There is a very strong correlate between TNF alpha and hostility. So I point that out to the patient. Almost every time they sit there and go, yeah, absolutely. Yeah, that's me. IL-6 tends to promote anxiety, and IL-10 tends to promote depression. So just looking at your detox panel, you can ask people, you don't have to tell them, hey, this is what it means, you're depressed. You can ask them what their mood level is, what their energy level is, and do you find that there tends to be melancholy or sadness or depression? And someone will say, yeah, I guess so. Okay, well, this is possibly one of the reasons why. This is your genes. This is your gene panel right here. This is what is written in ink, but just so you know, if we change some of your epigenetics, we can suppress this and we can express a better mood, but it's completely up to you if you want to put that kind of time and energy in. I find this to be very, very empowering for the patient. Multiple times they have said, oh my gosh, I thought it was just me. I just thought it was a lack of self-control. I thought it was just my personality, and I just tried to figure out how I'd be able to get out of this hole. But if you understand that the epigenetics would dictate that we can suppress this, that there is actually something that can be done, they get very, very excited about that. These are the pages that I tend to look at. These are the client summary reports. And you'll find over time, you'll be able to breeze through these pretty quickly, but it's still comprehensive. For one of the traits like the glutathione giving you the specific gene and specific SNP, it tells you what the significance of that SNP could be, what that variant type is, what that impact may be, and also what you'll find is under the comments, there are specific numbers for PubMed, and on that PubMed number, it will correlate to specific research. So if you just type that number in, you'll learn more about that specific SNP. And I just kind of nerd out on that stuff. So I like having that ability to say, let me know a little bit more about that. Let me see what that impact is about. Also in that page is nutrient recommendations in a summary form. So it will say, oh, for your inflammation pathway, for your inflammation trait, you might want to be on Inflamease or Herbal Ease and Methyl Complete, or if we know that there is a tendency to move towards the 4-hydroxyestrones, maybe move into an estro balance and dynamic hormone balance and cellular energy. Obviously, the new detox formula, if you haven't tried that, that's the initial go-to. So we have many options, and obviously as doctors and practitioners, we need to be able to make the final decision. But what I like about this report is when I see things mentioned multiple times, then I know it's something I need to pay attention to. Now, sometimes I might say, oh, well, there's some resveratrol, but I know there's resveratrol in the detox formula anyway. Or based on the person's symptoms, I'm going to do a detox formula and maybe the liposomal glutathione. So we have multiple options of what our protocols can and should be in the beginning with our maybe 4R program, all the way to wellness and sports performance. So that's going over the specific detox report and the information that we can glean from the reports, and how easy it is maybe to create a protocol. TIER is an acronym that I just made up, but we test, educate, and there's nothing better to educate the patients on what's going on with them than showing them what their genetic propensities are and how those genetic propensities have been manifested based on their lifestyle choices. So educate, and inspire them that they can make changes, and small changes incrementally over time can create huge changes in someone's life. And then we recommend a proper protocol, and I continue consulting with patients. First, I consult with them weekly, then I move out to every other week, then we go to every month, and then I like to check in once a quarter to make sure someone is following through with some sort of proper lifestyle plan. So that is the webinar, and I am open to any questions that anyone may have about the information or how I utilize the reports and how I discuss things with patients. So thank you very much for your attention, and I truly appreciate again your commitment to wanting to make your patients healthier. Let's see what we can do as far as questions.
So thank you, Dr. Scott, man. That was fantastic. What a deep dive into the detox panel. We do have a couple questions, so I can read them off to you if you want to answer them, and I think some of these I can answer myself. One is, let's see, with MyOrigin, are we able to get the raw data if we want to run it through additional analysis? I don't think so at this point. Jerry, I will have to get back to you on that.
If I can interject, one thing I didn't mention is that one of the options that you do have with MyOrigin, which is super cool, is you have these specific panels, but you can go in and add on. There's a way for you to do multiple add-ons, unlimited add-ons to whatever you want. So if I'll often do a detox panel, but then add on the MAO, which is also in the cognition, but instead of doing a full cognition panel, I just add that to my detox. If you want to make your own, or if you want the entire, whatever raw data there is in MyOrigin's database, that is available, but I don't think it's all several 100,000 SNPs, if that helps.
Yes, that does. One of the other questions was, there's a couple from Debbie. Not having a strong functional medicine background, can these still be applied to us practitioners who do not have that knowledge?
I believe so. I think with the reports where they're making general recommendations, I think it's a great first step. And some of the things that you looked at, they're making supplement recommendations, exercise recommendations, lifestyle recommendations, so and supplements. So there's a huge head start. There's a huge learning curve that you don't have to step into to run these reports. So they're very user friendly.
They are very user friendly, and the new reporting program or structure is so much easier to read. Another question from Kim is, do you have any examples of how you use the detox panel specifically with a patient, and do you recommend this panel first with a patient?
Good question. So we can run as many panels as we wish, right? I've had some people just say, yeah, let's do whatever you think is most important. I think the detox one is the number one. I think it gives us the greatest amount of information. Some people have just said, you know what, I don't want to fool around with this. Just run everything. I just want to see everything. So yeah, you can choose whichever panel. As you asked, I do use the detox panel primarily. That's my number one. And then if I think there's other things that I need, I'll pull those in. So you also asked, do I have examples of how I use this panel specifically? Yeah, I run my slideshow. I have my consult room. I've got a big screen TV. I've got my laptop hooked up to it. What's faded in the background right here with the phase 1, phase 2 detox, I pull that up and I educate the patient on what phase 1, phase 2 detox actually is. And I just go over the most basic one, not the one with the gene SNPs or anything like that. And I just tell them that there's molecules that get converted to something more toxic, and that more toxic molecule then gets surrounded by a water molecule, and then we eliminate it. Then I go through the specific genes that are phase 1 and phase 2. So I'll point out to them, you now have 7 SNPs on phase 1. So we're not even converting some of these metabolic toxins into an intermediate. You have 12 phase 2 SNPs, which means that we're not surrounding that more dangerous intermediate molecule into a water molecule. And if that keeps getting produced and not clearing, now we have all of these pro-inflammatory free radicals that are creating an inflammatory response. So we are going to first focus on your phase 1, phase 2 detoxification. Did I answer that okay? How I use that specifically for my patient?
Yep, I think that was great, Dr. Scott. We have another. We're going to take a live question here from Janice. Janice, I'm going to unmute your mic and go ahead and ask your question. You have to turn your mic on first, though. Maybe this isn't going to work. Yep, you're self-muted right now. If you want to turn that on, you can ask your question. While you're doing that, I'm going to go to the next question here, which is, do you have any idea how common some of these SNPs are? I have only run a few patients, and it seems like they were high risk for almost all traits. Is this common?
I am finding similarities with that. The SNPs are called variations or mutations because it's only about 1% of the world population of what they studied that have these variations. But realize that someone's walking into your office and willing to do this test because they're not feeling well. So the likelihood of them having a bunch of these SNPs is not uncommon. And remember what I said, we don't treat the SNPs based on someone's epigenetics, based on someone's lifestyle. Several of these SNPs might not be affecting us at all. But yeah, the first couple times I did the testing, it was a little shocking, like, oh my gosh, how am I going to tell this person that they're not dying tomorrow? So depending on which one, like a cardiometabolic one, most people with any kind of central obesity, most people who are stressed at all, they have several of those insulin resistant cardiovascular markers. It can be a little shocking for sure.
All right, and we have a couple other questions here from Debbie. How is the DNA collected from the patient? Debbie, we can send you a DNA collection kit. It has a cotton swab. It's a couple, I think it's 30 seconds on the sides of the cheeks, and then you have a little vial that you dip that in. It's a prepaid envelope right in there. You put it right in there, and you register patient on the MyOrigin dashboard through logging through Nutridyne, and you send it, drop it in the mail. And another one of your questions was, what's the turnaround time? Turnaround time can vary a little bit. Right now, it's between 6 and 8 weeks. And then another. Go ahead, Scott.
To piggyback on that, Joel, when I do my initial consult with a potential functional medicine patient, and I recommend the MyOrigins, I do it right then and there. The moment they say, yeah, that seems interesting, I don't have them take it home. We have a little area that we have for a lab. The kit is very compact, very easy. My staff actually brings the patient into the room. They rub that the applicator in the mouth. It's only like 30 seconds and it's done. And then my staff has the sample with all of the ID numbers, and my staff inputs and registers the person right then and there. So they walk out of the office at their consult, and it has really gotten to the point where the testing itself only takes about 30 seconds, and then my staff can input it later during lunch or during a break or something like that. So it's very, very simple, very easy for adults or children.
I agree. And Scott, do you find that having your office staff perform that procedure, you're getting higher compliance?
Yeah, whether it's my office staff or myself, but having them, if they agree to moving forward, there have been times, and it's just human nature. They take it home, and then little Johnny had to get stitches, and then, you know, something else happened, and you're 2 weeks in and they haven't even sent it out yet. And because the turnaround on the report takes the time that it does, we want to be able to capture that as soon as possible.
That makes absolute sense. And one of the last questions I have here, we have two more here. One is, what is the cost? So the wholesale pricing for the Healthcare Pro panels are $135, and it's manufacturer retail price is $159 to the patient. You guys have flexibility to do that. You can build in your consulting fee. You can give it to them for a cost, or you can give it to them for more. And then the nice thing we do is each panel after that you would want to run is only $35, or $59 to $159 to the patient. The Wellness Pros are $125 for the wholesale price. And what Scott was talking about earlier is you can add custom traits into your ordering at $5 each. So it's once you have that tested, you can really get granular on the testing that you're doing. It makes it very cost effective and an easy way to keep going back and getting more and more information. Looks like we have one final question. It's from Michael here. I'm going to unmute you, Michael. Give me 1 second here, and you can go ahead and go live if that will work. Go ahead.
Hey guys, can you hear me?
We can.
Just wondering where the lab is that does the DNA testing.
So we've partnered up with Toolbox Genomics as our partner lab. Scott, I'm sorry, I'm drawing a mind blank of where the lab is. Do you know, Scott?
Well, I know that their headquarters is in San Francisco, and I'm not aware of exactly which lab that they use.
Yeah, I just asked. I'm just wondering if this is specific for MyOrigin, or if this is a testing company that services other companies as well.
No, this is specific for MyOrigin. So, like I said, we have a back end company that we use, and they have specifically written these reports, and it's something that is just for this reporting.
Gotcha. Thank you.
You bet. Well, guys, we've reached our 1 hour limit. Dr. Scott, thank you so much for that wonderful information. I know it's a lot of information, everybody, and we appreciate you guys all hanging in with us. We are recording this, so it's going to be sent out to all of you as soon as the recording is processed. And you always can find more resources if you log into Nutridyne.com, click on MyOrigin, sign in, and you go to the resources page of the MyOrigin dashboard. You can find a lot of information there as well. Another really great resource that you have is your sales rep. Please reach out to your sales rep if you have any questions. If you have any urgent questions, you've already started the process of ordering kits and you have some urgent questions, you can email MyOrigin@Nutridyne.com, and that will get you a very fast reply. Again, thank you everybody for participating in today's meeting. Dr. Scott, it's always a pleasure. Thank you very much. And everybody have a great day.
Thank you.